Good morning, one and all. A warm welcome to all of you for the International e-workshop on Artificial Intelligence Tools for Accelerated Drug Discovery. Myself, I'm Rohini Krishna Kota, CSR SRF, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. I'm here to introduce our today's speaker, Dr. Ho Leong NG, Associate Professor, Department of Biochemistry and Molecular Biophysics, Kansas State University, Kansas, USA. Dr. NG is an Associate Professor of Biochemistry and Molecular Biophysics at Kansas State University. Dr. NG's training is in computational structural biology and crystallography. He was studied structure-based drug design since he was an undergraduate at Harvard University, studying under future Nobel laureate Martin Karplus. Dr. NG completed his PhD at UCLA, the University of California at Los Angeles, under the joint supervision of David Eisenberg and Richard Dickerson on crystallography and computational biology. He also completed 2 years in medical school as a part of his MD PhD program, which provided him a background in pharmacology. After UCLA, he worked as a postdoctoral fellow and staff researcher at UC Berkeley with Tom Albert. He then worked as a senior scientist at Conformed RX and reported directly to founder, future Nobel laureate Drain Kobilka on G protein coupled receptor crystallography. In 2011, Dr. NG moved to University of Hawaii as an assistant professor of chemistry. In 2017, Dr. NG moved to Kansas State University. Dr. NG is the recipient of US National Science Foundation Career Award for Early Career Scientists. Dr. NG has placed in the top 5 in several global computational structure-based drug research competitions. Dr. NG is the founder of Open Source COVID-19, a non-profit group dedicated to drug discovery and open science. Apart from this, he is an active and scientifically driven personality. Without further ado, join me in welcoming Dr. Ho. Sir, please.
Thank you very much. Let me get my screen started for you. How's that? Can you see that? Hello? Hello, can you see my screen?
Yes, sir. Yes, it's visible.
Okay, great. So let me tell you about our work on AI-driven drug discovery for COVID-19. Let me give you a little bit of a background on my research. So most of my lab works on structure-based drug design using both computational and experimental methods. We normally focus on cancer, immunology, and malaria, but with the great urgency of the COVID-19 situation, in February, we decided to also start work on COVID-19 and antiviral drug discovery. So this is quite new. This area is quite new to me, but when it comes down to atoms and molecules, they're all the same, right? They all have to follow the rules of physics and chemistry. I've been working on machine learning for drug design for about 4 years now, and I'm going to talk about how we can apply AI methods to accelerate and expand the power of modern computational drug design. So much of my work on open source COVID-19 has been inspired by my participation in open source Malaria. So I've been a part of open source Malaria for over 3 years now. This was one of the pioneering groups to do open source drug discovery. As many of you probably know, a lot of the earliest pioneering work was done in the area of malaria and tuberculosis in India. But the driving principles behind open source drug discovery projects are that we do not create intellectual property. So we don't make patents. All of our data and all of our results, and hopefully when we come up with actual drugs, will be available freely to the public. Another driving principle is that anyone, or at least any scientist, can participate. So the project and the data is not owned by any one person. So the organization of both open source Malaria and our organization, we try to decentralize as much as possible, and we try to get as much group feedback and public feedback as much as possible. With open source Malaria, led by Professor Matt Todd, they have now come to realize several very interesting potential drug leads, which we're still in the process of moving forward on this. But I want to emphasize this because we are not just discussion groups that are throwing around ideas. We're actually trying to really do drug discovery from biology to chemistry and ending with actual compounds. Let me give you an overview of the entire drug discovery pipeline. Normally, it can take 12 to 15 years to move from target identification to having an actual drug in hand that is approved for human use. Now, much of the early stage, of course, involves chemistry, and that is my specialty. And then the latter part of the process is focused on human clinical trials. That part can take a long time and is very expensive and very complicated because, of course, you must prove human safety and efficacy, and the work must be done by doctors and nurses and technicians in hospital settings. So this, of course, in many cases, this doesn't even work. So very often, the lead result of a drug discovery research project is, unfortunately, nothing. Most of the time, this fails. So this is what we call the funnel approach to drug discovery. We might start off in this figure with something like 10,000 compounds. So 10,000 compounds that might have drug activity in vitro. And we continue to sort through these compounds to find the ones that have the most promising properties for drug discovery and development, meaning that they would be biochemically active, they would be active at the level of cells, we can show activity in animals, and of course, again, we must show that these molecules are safe and non-toxic. So at the end, you start with, while you might start with 10,000, you might only end up with a handful of molecules that might make potentially good drug candidates. So this is a very difficult process. And like I said earlier, this can take over 10 years. And what I haven't shown here is that this can cost over $1 billion to drive this process. Now, this is something that is a huge problem because of the time and the expense. Now, in a case like COVID-19, many of the large pharmaceutical companies are not working on this. So that means that there isn't the kind of professional pipeline professional pipelines and teams for doing drug discovery. And also, we don't have the time to wait 10 or 13 years for a treatment for COVID-19. We need one as soon as possible. So how can we do this? So my approach and that of many of my colleagues in this field is we're making a strong focus on CAD, or computer aided drug discovery. We hope that by using the latest advanced methods developed by our colleagues and ourselves, that we can accelerate the early stage chemistry to get molecules that are more likely to be successful in human trials and more likely to be safe. And of course, using the latest AI methods is a big part of this. So let me just tell you very briefly about open source COVID-19, our group. So this is we are nonprofit open source. So data and results will be freely available. We want to lower the cost of drug discovery, and by removing the intellectual property aspects, we think we can do that as well as accelerate it. We are inclusive and we are open, and we welcome new scientists. We have many members from around the world, with many uh, in uh, in developing nations. We have almost all capacities for doing work on drug discovery, from computational drug design to chemical synthesis, biochemical testing, protein crystallography, and testing with live virus. And that is our immediate goal, is to develop compounds by the end of the year that will be ready to go into, or perhaps even pass, safety and effectiveness trials in animals. We are currently still looking for new members, especially in the areas of chemical synthesis, and of course, for partners who can support us financially, because although we are all doing this for a nonprofit, we still have to pay the bills for research services and reagents. So please, if you know people who want to join us, let them know about our group. So in our group, we're focusing on we're working on about uh, 4 different targets in SARS-CoV-2, but the 2 that we're the most advanced in are M-Pro protease and NSP16, which is an RNA methyltransferase. Now, M-Pro protease is currently the most heavily studied drug target in COVID-19 research. We and many other groups, both in the biotechnology industry as well as other academic